Today's webinar, From Biology to Decisions, Designing NAM Workflows to Build Scientific and Translational Confidence. It's being presented by Drs. Kent Grindstaff and Dr. Bhagwat Prasad. Introduce them both in just a moment. I'd like to introduce you to our first presenter, Dr. Kent Grindstaff, who's the Consulting Director of Scientific Affairs at BioIVT. And Dr. Grindstaff brings more than 25 years of experience across multitude of biotechs, pharmaceutical, and CRO environments, working on drug discovery, translational research, preclinical development. He's led cross-functional research teams, advanced therapeutic programs across multiple disease areas, and supported biomarker and regulatory strategies. He's earned a PhD in molecular cell biology and biochemistry from Washington University and completed his postdoc training at Stanford. At BioIVT, his work centers on fit-for-purpose study design and translational workflow integration, which is at the heart, I guess, of today's discussion. And our second presenter is Dr. Bhagwat Prasad, who's at Division Director of Translational and Clinical Pharmacology at Cincinnati Children's Hospital. And he's a professor at the University of Cincinnati Department of Pediatrics. Dr. Prasad's research spans drug metabolism, disposition and safety, quantitative proteomics, metabolomics, and biomarker discovery. I hope I got those right, with a particular emphasis on improving drug safety and efficacy for children and other populations underrepresented in clinical trials. He earned his MS and PhD in pharmaceutical analysis from the National Institute of Pharmaceutical Education and Research in India, and completed his postdoc training at the University of Washington. So we're very pleased to have both of them here with us today. So bear with us, and we'll get things over to Dr. Grindstaff to begin the presentation.
All right, Tom, thank you for the introduction, and thank you to NJABR for inviting me to join you today. What I want to discuss is how we think about new approach methodologies, or NAMs, as part of the broader drug development workflow. There are obviously many exciting technologies within the NAM space. We've advanced cell culture formats, microphysiological system, ex vivo approaches, computational models, as well as in vivo, and a growing range of analytical tools. But I'm not going to focus today on any particular technology. Instead, I want to take a step back and look at the workflow around implementation of these technologies. The title of the presentation is From Biology to Decision, and that's really the organizing theme for the talk. At the end of the day, the value of an experimental model is not determined by how novel it is or even how biologically sophisticated it appears. Its value depends on whether it generates evidence that helps us answer a specific scientific question and make more informed decisions. That means thinking intentionally about the entire path, from the biology entering the experiment, the model format, the endpoints and analytics, and ultimately the interpretation of the evidence. I'll spend a little more time on biological starting materials because I think they're an important and sometimes underappreciated part of model performance. But the broader point is really around intentional workflow design. And towards the end, I'll bring that back to how NAMs and in vivo models can be integrated, what is required for these approaches to become more reproducible and scalable, and then hand the discussion over to Bhagwat. He'll take one important aspect of that further and address quantitative proteomics and biological confidence. Now, NAMs are often discussed in the context of reducing or replacing animal studies, and that's certainly a driver of the field, and is part of the regulatory and policy momentum we're seeing today. But scientifically, I think that framing is too narrow. There are many questions for which in vivo models remain extremely valuable. They can provide insights into integrated physiology, whole organism exposure, interaction among multiple organ and biological systems, and a systems level context that can be very difficult to reproduce in a more reductionist model. In many areas, they also bring decades of historical experience that help us understand how to interpret a result. So this is not an argument that legacy models no longer have value. At the same time, we know that no individual model captures every aspect of human biology. Species-specific biology can create translational gaps. Some mechanisms can be difficult to isolate within an intact organism. The diversity present across human patients may not be adequately represented. Exposure response relationships don't always translate cleanly between species, and highly targeted specific therapeutic modalities, there may be aspects of mechanisms that simply are not well represented in traditional formats. This is where NAMs can add something important. They provide additional ways to generate evidence. Sometimes they allow us to isolate a particular mechanism more precisely. Other times they allow us to introduce human-derived biology directly, and they make it possible to examine variability across donors or disease states. But in many cases, the greatest value may be complementary. A NAM may help explain a finding from an in vivo study. An in vivo study may provide physiological context around a mechanism first identified in vitro, or multiple evidence sources may converge and collectively provide stronger support than any single model could provide on its own. That distinction is important because if we begin by asking, can this NAM replace the animal study? We've already framed the problem as a competition between model formats. I think the more useful starting point is the statement at the bottom of the slide. What evidence is needed to address scientific questions and inform the decision? Once that's clear, we can identify the gaps in the evidence and then determine which tools are best positioned to fill them. That might be a NAM, it might be an in vitro model, or some combination of approaches. So throughout the talk, when I refer to NAMs, I'd like you to think of them primarily as evidence-generating tools within a larger scientific workflow, not replacements by definition. Now, there's clearly a great deal of momentum around NAMs right now. The technology themselves are becoming more mature. There's a greater regulatory openness to considering these approaches. Industry investment is increasing. We're seeing more collaboration among pharmaceutical companies, regulators, model developers, academic CROs, as well as the groups that are building the analytical and computational tools around these systems. And importantly, the conversation is beginning to move beyond individual technologies toward how different types of evidence can be integrated. All of that creates real opportunity, but it also creates a potential challenge. Enthusiasm for technology can sometimes become confused with confidence in the evidence. They're not the same thing. A model does not become more informative simply because it's new. It's not automatically more predictive because it's non-animal. And greater technical sophistication doesn't necessarily mean greater biological relevance. You can have a technically elegant system that's poorly aligned with the scientific question. Conversely, a relatively simple experimental system can be extremely informative if it captures the exact biology required to answer that question. So while the landscape is shifting, the fundamental scientific standard is not. The approach still has to be justified. We still need to understand what biology is representative, how consistently the system performs, what the limitations are, and what conclusions the data can reasonably support. And I'd argue that those considerations become more important as the range of available models expands. Today, we can choose among cell lines, primary cells, co-culture, spheroids, organoids, microphysiological systems, ex vivo tissues, in vivo models, and computational approaches, as well as combinations of all of these. That breadth is a major advantage, but it's only an advantage if we have a disciplined way of selecting among those options. So the real opportunity is not simply that we now have more models, it's that we have more ways to construct evidence packages. And that means the scientific rationale for choosing a particular approach becomes even more important. That brings me to what I think is one of the most important principles in NAM's implementation, and that's start with the decision, not the technology. It sounds obvious, but in practice, it's very easy to do the opposite. A new platform becomes available, we see a compelling publication or conference presentation, we're interested in the technology, and the first conversation becomes, where could I use this? That's understandable, but it's technology driven rather than question driven. The starting point should be the decision that the evidence is tended to inform. Underneath that decision is the scientific development question, and somewhere within the existing body of evidence is often one or more gaps that prevent us from answering that question with sufficient confidence. Once those pieces are clear, the model has a defined job. It's no longer being used simply because the platform's available. It's being selected because it's expected to generate a particular set of evidence. That's where context of use becomes extremely valuable. The context of use should make clear what question we are addressing, what decisions the evidence is intended to support, what gaps, if any, exist in the current evidence, and what level of performance we need from the model and who it's intended to inform. That context begins to shape the model selection. If the problem is primarily mechanistic, we may actually benefit from a relatively controlled system where individual variables can be isolated. If the question depends on interaction among multiple cell populations, then we may need additional biological complexity. And a scientific question depends on understanding variation across patients, donor diversity may be far more important than adding another level of engineering sophistication. The same logic applies to endpoints. We should not simply measure what is convenient or historically familiar. The endpoint need to be sufficiently connected to the biological mechanism and the intended decision so we can interpret what the results mean. Sometimes that means a functional endpoint, sometimes it means molecular characterization, but often it means combining several types of measurements so that we understand not only whether something changed, but why it changed. The analytical strategy should follow the same principle. More data does not automatically create better evidence. A very large data set can still leave the central question unanswered if the measurements are not aligned with the intended decision. The 4 prompts in the center of the slide are there less of a checklist than a way of keeping the experimental information anchored. We need to know the scientific question and the evidence gaps. We also need to understand who ultimately has to rely on the results, whether that is an early discovery team, a development team, or a regulatory reviewer, because the depth of characterization that's needed may differ substantially. And finally, the experiment should connect to an action. If a positive result and a negative result would lead to the exact same next step, then the study may be scientifically interesting, but it's probably not decision relevant. That's why I think the intended decision belongs at the beginning of workflow design rather than at the end of data generation. So once the question, evidence gap, and intended decisions are clear, we can begin to design the workflow around those needs. And although this slide sits within a NAMS discussion, the logic itself is not NAMS specific. This really is a framework for good experimental design in general. The first component is the input biology, what biological materials entering the system. That includes the donor or specimen, the relevant metadata or provenance, and the biological variability that may matter for the application. Those choices establish the biological starting point for everything that follows. The second component is the model itself, the format and the level of complexity needed to match the question. And there's an important balance here. Every added feature can potentially contribute additional biological information, but each addition can also introduce new sources of variability, technical failure modes, cost, throughput limitations, or analytical complexity. So the goal is not to construct the most complex model possible. The goal should be to build a system that's sufficiently complex to answer the question and no more complex than is necessary. Then we come to endpoint analytics. This is where we evaluate the model is actually going to tell us. The endpoint should reflect the biology we need to interrogate and should be aligned with the intended decision. A viability readout may be perfectly appropriate for one use case and completely inadequate for another. And a functional endpoint may be important if we need to demonstrate biological capacity. And in many situations, combining different types of measurements gives us much more interpretive power than relying on a single endpoint. Then we have characterization and controls, which is intentionally shown as spanning the entire workflow. I think this is an important point because we sometimes talk about model characterization as though it's a separate activity performed after the model has been developed. In reality, characterization begins with the inputs. We need to understand the identity and quality of the biological material. We need to know whether the model performs consistently. We need to know whether the endpoint itself is reproducible, and we need to understand the boundaries of interpretation. Every experimental model has boundaries. Still, that's not a failure of the model. A highly controlled in vitro system may be intentionally set up to omit features of systemic physiology, and an animal model may not reproduce important aspects of human-specific biology. Those limitations become problematic only when they are poorly understood or ignored. A well-characterized model can be extremely useful within a relatively narrow context of use. A poorly characterized model can create false confidence even when the platform itself is technology impressive. The final part of the slide is the transition from individual measurements to integrated evidence. Major development decisions are rarely made from a single isolated result. We typically consider several pieces of evidence together and ask whether they support one another, whether they conflict, and whether the overall evidence is sufficient for the decision. NAM should fit into that same logic. The context of use provides the anchor for the workflow, but the workflow remains iterative. If the data reveal that donor variability is dominating the response, we may need to refine donor selection or intentionally broaden the donor population. If the model doesn't reproduce the expected biology, the format may need to change. Those adjustments are not evidence that the workflow failed. They're just part of the normal process of developing an experimental system that's actually fit for its intended purpose. And I think that distinction is important. There's a difference between demonstrating that NAM technology can generate interesting data and building a NAM workflow that consistently generates interpretable evidence. The technology is one component, but the workflow is ultimately determines whether evidence can support a decision. That brings me to part of the workflow I want to discuss on the next few slides, the biological inputs. The simplest way to make the point is that biological material are not neutral reagents. Their identity, their state, their composition, their variability influence what the experimental system can represent before we ever begin working on the model itself. At a high level, there are three ways this can become problematic. The first is biological mismatch. The material may be technically high quality, but it may not represent the biology relevant to the question. The wrong disease state, an inappropriate cell population, or the absence of biologically important characteristics can functionally limit what the model is able to tell us. The second is unresolved biological variation. We may observe variability in experimental output and assume it reflects poor technical performance, when it may actually represent meaningful differences among donors or specimens. That distinction matters. Biological variation is not simply noise that should always be eliminated. In some applications, understanding that variation is actually part of the scientific objective. And the third issue is material integrity. Collection, processing, storage, specimen composition, and other upstream factors can alter the biology before the material ever enters the experimental system. Once that biological state is changed, no amount of downstream analytical sophistication can reconstruct information that was lost upstream. So before we ask how sophisticated the model is, there's a more basic issue to resolve, and that's what's the biology we're actually putting into the system, and is the biology we intend to study. When I talk about starting material quality, I'm not suggesting that there's one universal definition of high quality sample. In fact, that conflicts with the context of use framework we just discussed. Quality has to be considered relative to what the material need is intended for in the experiment. The first issue is representation. The biology we select needs to reflect the scientific question. For a disease model, that may mean a particular disease state, phenotype, treatment history, pathology, or cellular composition. For another application, functional capacity may be more important than disease state. And if the objective is to understand population variability, then a highly homogeneous set of donors may actually be the wrong experimental choice. So sourcing is more than obtaining the correct tissue or cell type. It means selecting biological material that represents the context we're trying to model. The second consideration is biological variance. We often use the word variability as though it's inherently undesirable. Technical variability usually is undesirable, but biological variability can be informative. Patients, donors, disease states, cellular composition, expression levels, functional capacity can all differ. The relevant question is whether those differences matter for the intended use of the model. If we're trying to isolate a specific mechanism, we may deliberately control the biological background as tightly as possible. If instead we're trying to determine whether a finding generalizes across the patient population, then including biological diversity becomes essential. Those are very different study designs. The third element is characterization. The level of characterization should always follow the scientific need. Not every specimen requires every possible measurement, but if genotype affects a particular pathway we're studying, then genotyping becomes important. If tissue composition can alter the endpoint, pathology may be essential. And if we need to understand how a model compares biologically with human reference tissue, then molecular characterization may become an important part of the evidence package. The same principle applies to collection and processing. How quickly was the material processed? What happened between collection and cryopreservation? Were handling conditions consistent? Is the biological state we care about likely to have been preserved? Those may initially sound like operational details, but they become scientific variables as soon as they alter the phenotype or function in the model format that we're looking at. Traceability is equally important. If we can't connect the experimental results back to the donor, specimen, collection conditions, processing history, and relevant metadata, it becomes much harder to understand why the model behaved the way it did. That's why I like the concept at the center of the slide. Starting material is a quality system. It includes sourcing and biological representation. It includes collection and processing. It includes the characterization and includes traceability and documentation. Still, the required depth of each component depends on context. There's another statement on the right side of the slide that I think captures the overall point particularly well, that downstream analytics can quantify the output, but they can't compensate for an experimental system that does not adequately represent the relevant biology. We can measure the wrong biology very precisely, but that doesn't make it the right evidence. And that's why I think starting material belongs within the evidence strategy rather than being treated as simply background reagents. They help to define what the model is capable of representing and therefore what conclusions can reasonably be drawn from it. So I'll use two relatively simple examples to make that point tangible. The first is donor variability data from leukopak collections at our donor centers. I'm not going to walk through the individual markers here. The overall pattern is what matters. Across the immune cell population shown, variability between donors is substantially greater than the variability observed within the same donor on repeat collections. Depending on the marker, the difference is roughly 2 to 5-fold. And as you can see in the graph on the right, some NK markers like CD56 show particular high interdonor variability. The important measure is that the donor itself is an experimental variable. If the behavior of the model depends on the abundance or state of a particular immune cell population, changing donors may create meaningful changes in the biological system. So when an assay behaves differently from one experiment to another, we shouldn't automatically assume that that's a difference in technical noise. Sometimes the biology is changed based on the input material. And if the biology variability matters for the context of use, donor selection, donor characterization need to become part of the experimental design. In the next example, the same principle applies at the specimen level. A clinical diagnosis tells us what disease the patient has, but it doesn't necessarily tell us exactly what's present in the particular section of tissue used in an experiment. Tissue regions can be highly heterogeneous. A single surgical specimen may contain tumor cells, adjacent normal cells, fibrotic tissue, inflammatory infiltrates, necrosis, and other features that are very different in proportions. So two blocks from the same specimen can produce very different molecular and functional results because they contain different biology. If the interpretation depends on tissue composition, pathology review can help confirm that the specimen actually represents the biological context we intend to study. Again, the point's not that every sample needs the same level of pathology characterization. The point is that the quality control should also be fit for purpose. If specimen composition can influence the conclusion, we need enough information to understand the composition. These are simple examples, but together they reinforce the same idea, that the performance of the model can't be separated from the biology entering it. So once we understand the biology that we need to represent, we come back to the model selection itself. The principle here is straightforward. Complexity adds value only when it improves interpretation for the intended use. And I want to be careful about the way the model formats on the slide are interpreted. They're not intended to represent a universal hierarchy from simple to complex. And furthermore, biological complexity and technical complexity are really different dimensions of the models. So I suggest thinking about these as different ways to represent biology rather than steps on a ladder. A 2D culture may be ideal, where we need a scalable functional or mechanistic measurement with relatively tight experimental control. And a 3D model system can add features such as spatial organization, gradients, matrix effects, and other aspects of the tissue. Organoids and MPS formats may allow us to reproduce tissue features, define cell-cell interactions, or exposure dynamics that are difficult to capture in 2D systems. And an ex vivo tissue can preserve native architecture, heterogeneity, and disease context that may be difficult to reconstruct experimentally. None of these features make one category universally better than another. The model has to be selected according to the biology required by the question. For me, there are three useful considerations here. The first is mechanistic fidelity. Does the system include the biological components necessary to interpret the signal we are measuring? The second is exposure relevance. Does it reflect the tissue context, dose, duration, exposure behavior necessary for the question? And third is decision relevance. Does the added complexity actually improve the evidence or provide the evidence we need for the intended decision? Because complexity has a cost. It can be biologically informative, but it can also reduce throughput, increase variability, complicate analysis, and introduce new failure modes. So there are two guardrails. We don't want a model that's so simple that it misses the essential biology, but we also want to avoid complexity creep. Adding features simply because they're technically possible rather than because they materially improve the evidence shouldn't be the approach either. The objective is not to maximize complexity, it's the right biological complexity for the context of use. This brings us back to the framing from the beginning of the talk. If different models capture different aspects of biology, then the objective does not necessarily need to be choosing one and eliminating another. The more useful approach is to understand what each evidence source can contribute to the scientific question. In vivo models can provide integrated systemic physiology, whole organism context, and in many cases add established body of historical or regulatory evidence. NAMs may allow us to define experimental conditions more tightly, integrate a particular mechanism, or introduce human-derived biology directly into the workflow. But I want to be very clear that those are illustrative capabilities, not fixed advantages. There are in vivo models that provide excellent mechanistic information. There are NAMs with substantial biological complexity and variability, and not every NAM uses human-derived biology, and not every in vivo model provides all of the integrated information required for a particular question. The value depends on the specific model and its context of use. More importantly, every model can leave gaps. Recognizing those gaps changes the way we think about integration. If an in vivo study identifies a biological effect, but the mechanism is unclear, a more defined NAM may help separate the underlying process. Conversely, if a NAM identifies a mechanism under tight controlled conditions, An in vivo study may provide information about whether that mechanism remains important within integrated physiology and realistic exposure. Sometimes the evidence converges. That convergence can strengthen confidence, and sometimes the results disagree. That doesn't mean that one model is wrong. The disagreement may expose an important species difference, a difference in exposure, an effect of model composition, or a boundary in the biology being represented. In other words, the disagreement itself can be informative evidence. This is why I think integrated evidence works best when it's intentional. If we understand from the beginning what the question each evidence source is intended to address, the studies can be designed to complement one another rather than producing separate data sets that we only try to recognize afterwards. This is the meaning behind the statement at the bottom of the slide and noted at the top. Not replacement by default, but complementary evidence by design. The goal is not to defend one model category. The goal really should be to construct the strongest scientific evidence package for the decision we need to make. The next challenge is moving from a model system that provides promising results in a defined study to a workflow that can be used routinely in drug development. That transition is often harder than it first appears. The model can perform very well in a proof of concept setting. The developer can understand the system intimately, the biological material can be carefully selected, the protocol has been optimized, and operators know which steps are sensitive. But routine implementation introduces a different set of expectations. The question and intended decision have to remain clear because, well, those established performance standards are essential for the workflow. Without that context, it's difficult to know whether variability is successful, whether a particular endpoint is sufficiently informative, or whether the model is actually performing well enough for its intended use. The second requirement is technical reproducibility. Can the experiment be repeated with sufficient consistency performance? That doesn't mean that every biological result needs to be identical, but it does mean that there's an important issue with the technical component that variation is understood and controlled well enough that meaningful biology differences can still be interpreted. Then we have biological benchmarking. This is a distinction from technical reproducibility. A model can be technically reproducible and still fail to represent the biology we care about. So we need some way of understanding how the outputs relate to known biology. That may involve comparison with reference tissue, known positive and negative controls, clinical observations, or other established experimental systems. The goal is to establish that the model is not simply consistent, but that the consistency is occurring within an appropriate biological context. The fourth element is operational scalability. Can the workflow move beyond the hands of one person who developed it? Can another operator reproduce the results? Can the necessary biological materials be obtained with sufficient consistency? And can the results be analyzed in a way that remain interpretable as the volume of experiments increase? Those may sound like operational questions, but they have direct scientific implications. If the model behaves differently every time the operator changes, we need to understand why. If a critical biological input can't be sourced consistently, that may define a practical boundary for the model. And if the analytical burden is so high that the workflow can't realistically be deployed, then even scientifically strong data may have limited impact in routine development. Finally, there's this decision integration. The result has to be framed in a way that allows people to act on it. Teams need to understand what a positive result means, what a negative result means, and where the interpretation becomes uncertain. They also need a way to interpret situations where the NAM result disagrees with another evidence source. This is where a lot of promising applications struggle. They're developed around a successful experiment rather than around a repeatable workflow. The original data may be excellent, but if the characterization needed to support use across inputs, operators, sites, programs, stakeholders was never really designed into the system, it may not be scalable. So when I use the term scalability here, I'm not simply talking about throughput. Scalability means the ability to reproduce, compare, interpret, and ultimately defend the evidence as the application expands. And that's why characterization and benchmarking need to be built in early rather than added only after the model begins to move towards broader use. So before I transition to Bhagwat, I want to bring back the discussion to 5 principles. The first is NAMs are evidence tools, not simply replacements. Their value comes from addressing a defined scientific, mechanistic, or translational gap. Second, start with the intended decision. The context of use should guide the biological inputs, the model system, the endpoints, and even the level of evidence expected from the workflow. Third, treat the starting biology as a part of the model. The cell, donor, or specimens are not simply source material. Quality, metadata, variability, pathology, function state, and traceability can all affect what the model is capable of representing. Fourth, match complexity to the scientific need. More complexity is not inherently better, and complexity, when it improves interpretation for the intended use, should be employed. And finally, build the evidence or confidence narrative from the beginning. Characterization, reproducibility, comparability, limitations, and biological benchmarking should not be questions we ask only after the data are generated. They're part of the experimental design. If we approach NAM development in that way, I think the conversation becomes much more productive. It moves away from debating which technology is best or whether one model should universally replace another, and it moves towards the much more useful question of how we intentionally construct the evidence needed for the scientific decision. That's ultimately what builds scientific and translational confidence. So intentional workflow design also requires tools that help us understand whether biology within those systems remain appropriate as we move forward towards translation broader use. That includes tools that allow us to use characterize biological capacity, benchmark systems against one another, and strengthen reproducibility and scalability. Quantitative proteomics is one example of this, providing a pathway to molecular characterization, capacity, and comparative biology across tissues, donors, models, and experimental systems. So rather than getting ahead of the discussion, this is a good point for me to turn things over to Bhagwat, who's going to take us much deeper into how quantitative proteomics can support translational confidence across workflows.
Okay, wonderful. Yeah, thank you, Tom, and thank you, Kent, for a wonderful, the first half of the presentation. And welcome, everyone. Good afternoon, good morning, wherever you are. I would also like to thank NJABR for this opportunity to basically highlight how quantitative proteomics can be used in characterizing new approach methodologies and complex in vitro systems. What I'm going to share in this particular talk is more examples, as Kent has beautifully covered the theoretical part of the NAM characterization and the importance of control and rigor and characterization of these models. So before I proceed, I have a conflict of interest disclosure. So I'm a co-founder and CSO of Quantomics. And my perspective today is centered on how rigorous protein measurement can strengthen confidence in NAM-based decision making. So I would like to start with a very famous statement here that all models are wrong, but some are useful. And this is really important that we're talking about the context of use in case of NAM, that some of them are useful for a particular context of use. As we know, many in vitro models or NAMs fail not from the model platform itself, but from inadequate biological characterization, as Kent highlighted. In my presentation, I will be highlighting how proteomics, and particularly quantitative proteomics, can directly test whether the molecular machinery required for example, toxicity assessment or drug metabolism or disposition assessment is present in a clinical relevant level in these models. I would like to start with this example and want to highlight why it is important to quantitatively characterize proteins in these models. This is an example of HepG2, again, which is not a NAM anymore, but it used to be a NAM when we started, cell culture and the hepatic cell line. And at that time, if we don't understand that the drug metabolism or drug metabolizing enzyme levels is extremely low or almost insignificant in this model, we would be using it without knowing the consequences, particularly when it comes to tox studies. If drug metabolism is not present, the intercellular concentration would be very different of drug or toxicant that can lead to misinterpretation of data. So what I'm showing here is these blue dots are basically the protein abundance of drug metabolizing enzymes in HepG2 cell as compared to primary hepatocytes, which has variability but way higher in abundance as compared to what we see in case of HepG2. So what we believe is that NAM is an opportunity, and it's an excellent opportunity, as Kent also highlighted. But when it comes to regulatory acceptance, it requires that quantitative confidence, quantitative evidence of human relevance. And what I will show is the proteomics. It will be a tool, one of the tools that will provide that measurement, which will help us basically translate and use these data for a particular context of use. So briefly about omics and NAM characterization. So one can use all different kinds of omics tools, including transcriptomics, proteomics, metabolomics. We leverage quantitative proteomics because most of the time the quantitative proteomics is basically telling you about the function. And in comparison with mRNA, for example, here what I'm showing is the mRNA does not mean protein. And this happens a lot when you're dealing with a very dynamic systems like these new approach methodologies, where the mRNA expression can be changing in a very different speed as compared to protein expression, right? So as you know, that the mRNA half-life could be in hours, but the protein can have a half-life of days to weeks. And that can cause a very different mRNA to protein expression differences in these models over time. This example in particular is just showing in primary human hepatocytes how protein expression changes in relation to mRNA expression changes. And as you can clearly see here, that mRNA is very sensitive. You will see changes happening within 24 hours. However, the protein differences are not evident in this particular case. So it's really important that when you're using this in vitro model, it should have the protein expression of the drug metabolizing enzymes or transporter or drug receptor or for a particular context of use. So why we believe the proteomics is important for NAM to human translation, because what you can do is in proteomics you can measure proteomic abundance in the NAM model, this in vitro model, and you can also measure the same thing in human tissue, and that will allow that link and scaling between human tissue and the in vitro models. Of course, activity assay can be done using the in vitro model, but oftentimes we can't use in vivo systems for activity assays, particularly for, say, transport assay or toxicity measurement. Also, when it comes to feasibility of the translation, we can clearly see that the protein-based characterization will help us this translation as in drug metabolism or drug clearance and disposition. The clearance intrinsic certainly depends on the expression or abundance of that particular enzyme or transporter. So here is what I was showing in the previous slide, that in biochemistry, the clearance intrinsic is directly proportional to the Vmax. As we can see here, the clearance intrinsic depends on Vmax divided by Km, but directly proportional to Vmax, and Vmax is directly proportional to the enzyme abundance. So as enzyme abundance changes, the protein activity will change accordingly and in a proportional manner. So we believe that the protein quantification can help us assess this gap between in vitro and in vivo system, similar to what it helps in understanding inter-individual variability. So there are a number of factors which can affect inter-individual variability across individuals. And we use enzyme abundance to understand it, and we basically develop successful PBPK models or QSP models based on these enzyme abundance differences. So the question is, how do we do proteomics? And so this is a very simple, I will not spend much time here, but what we are measuring in proteomics, in quantitative proteomics, is peptides. And these peptides are measured as surrogate to quantify the digested proteins. And so we can use a tissue, or we can use a NAM model or homogenate extracted from the NAM model, or the media and extracellular vesicles isolated from media. We can digest this and quantify. Both we can do, for example, targeted proteomics or global or untargeted proteomics. And what it gives you is it basically provides you a large number of protein data in quantitative manner. And so you can do multiplex quantification, which can be then compared with human tissues, for example, if you are doing with the NAM model. So how can you apply this in NAM characterization is one can leverage this to understand the difference between the NAM model versus the human tissues, right? So if you're dealing with drug metabolism, how much CYP3A4 expression is there in my in vitro model as compared to human tissue when you use the same amount of total protein? How is the protein amount changing over time? So looking at the temporal consistency of proteome. Also, one can leverage in characterizing cross-species scaling and characterizing these NAM for across species. Are they relevant to human, for example? Also, capturing variability. So, especially when you're deriving from primary cells, if the primary cells are retaining the consistency in terms of the proteome of a particular individual, we can use proteomics data to understand variability in the system. And finally, these data can be integrated into PBPK model in quantitative systems pharmacology models or toxicology models. So here is an example of the proteomics data that Precision Quantomics generated. This is basically quantifying human proteins in 40 different human liver samples. This is homogenate. And what I'm trying to show here is the consistency of proteomics data. So as you can clearly see, the gap pH in all these samples are not much different across samples. But then when you look into CYP3A4, this is where Kent was highlighting the biological variability. You can see huge variability in these enzymes. And that's what we need. We need to see how variable the protein expression of interest is, when it comes to drug metabolism or drug transport, as compared to the housekeeping protein or as compared to the whole proteome, which will be then relevant to see whether we can compare it with the across populations. So the same concept we can apply to NAM characterization. So you can think it as different batches of NAMs, and if you have a confidence that these are consistent expression of housekeeping protein, and we can see changes in the protein abundance of the protein of interest, we can characterize that model efficiently. So another question is, does this protein expression matches with the activity, right? And so we believe, and we have done consistently multiple experiments and shown that protein expression directly correlate with protein activity. And so when it comes to drug metabolism, we have clearly shown that the drug metabolism activity correlates beautifully with the expression, depending on if you have probe substrates that are very sensitive and selective. So most of the time it depends on the probe substrates rather than the protein expression. Of course, you need high quality data for proteomics as well, but you can clearly see that the protein expression does correlate with the activity. And so the proteins that we deal with in in pharmacology are drug metabolizing enzymes or transporters, or different binding proteins, and they are highly variable. And so understanding their non-conserved nature, that they are highly variable, and our body can withstand that variability. There is no clear phenotype when CYP3A4 is tenfold variable from one person to another person, except when you take a medicine. And so because of that, biology can be deceived by different level of protein abundance without having any physiological impact, except when you take a medicine or when you're exposed to xenobiotic exposure. Here is an example of UG2B17 that in biology, these proteins can vary significantly. We have individuals, almost 25% of patients do not have this particular gene. This is a drug metabolizing enzyme. And we don't have a clear phenotype in these gene deletions. So it's a non-lethal genotype. There is no clear phenotype in this particular case. In male versus females, also we have huge difference, almost four-fold difference. And then also we have seen very interesting ontogeny, so the age-dependent differences in the protein abundance. So what I'm trying to highlight is these proteins are highly variable, and when you are developing a NAM model for these proteins, you're going to see that difference without having any apparent difference in the cell type or cell physiology, cell viability, right? So the viability of the cell will not be different, but your CYP3A4 level could be tenfold different. And that's where characterization of protein abundance becomes extremely important. So the good thing about, so I was basically showing this slide where you can clearly see the difference in UG2B17. So the good thing about the proteomics data is you can integrate these data into PBPK modeling. So what we do in case of age-dependent variability in protein abundance, we can integrate this into physiological model, and we can predict drug clearance based on the changes in the level of these proteins that are determining drug metabolism or drug transport. And the same concept can be applicable to when it comes to in vitro models. So you can have NAM data, and that NAM data can be integrated into these models. So we can do NAM to human translation the same way we do inter-individual variability scaling. So another thing I want to highlight is we have developed in Precision Quantomics this translational proteomics atlas, which is allowing us to integrate these data and compare these NAM data with the human liver, human organ data, for example. So here is a case of human liver samples. So these individual dots are individual human liver samples. We have basically looked at, this is a case of ESGR1, for example. And what we are highlighting here is their distribution in this age versus their protein abundance in this distribution map. And what we then compare is, this is our HAPRF, which is kind of the pooled sample that is showing the expression right in the middle here. So what we can do is we can compare with this the HAPRF or the reference product, these 3D organoids. So these are 3D organoids derived from primary cells, and we can develop these human relevance scores. So with the PQTP atlas, we can generate the human relevance scores, and this human relevance score will allow us to see how close they are in terms of at individual protein level or individual pathway level, and how functional, whether the functional markers are, all functional markers are present there or not, and can basically advance this NAM characterization through human relevance score. So with this final example, I just want to highlight that authentication of these biological reagents and NAMs is essential for reliable in vitro to in vivo extrapolation and predictive toxicology. Quantitative proteomics, I hope I have convinced that will provide or is providing data that is allowing us in terms of precise multiplex measurement of admit proteins that can help us validate these the biological fidelity, detect donor-to-donor differences in these NAM models, and also strengthen the lot selection when we are developing these different lots of cell types or different cell lines, for example. When integrated in PPG model, these proteomics data can improve predictable accuracy and support precision medicine. So finally, I will like to close this talk by saying that the measurements defines this truth, and the quantitation of these proteins is helpful in defining this trust in this case of NAM model. And this is the guiding principle behind the PQ's work that I presented. With this, I would like to acknowledge my institutes and the collaborators who basically did this work. So from Cincinnati Children's, from Precision Quantomics, and also collaborators at Altis, and also PRINCE Consortium, that is a non-for-profit consortium within my lab at Cincinnati Children's. So with that, I would conclude my talk and happy to take any questions during the question and answer session.